Welcome grade 6 to your eLesson, chapter number 5, Plants and Photosynthesis. Today we are going to read page number 70, 71, 72 and 73. So starting the page 70, the starch test. An easy way of finding out if a plant is photosynthesizing is to see if its leaves have produced any starch or not. So what you need to do is you have to conduct an experiment to see if the plant is making its food or not. Before starting any experiment, you need to take your precautionary measure, that is, remember to wear safety glasses. To conduct the experiment, first point is that put a leaf in the boiling water for about 2 minutes to soften it. Number 2 is heat the leaves in alcohol to remove the green color. The flame must be put out, alcohol burns. Third point is soften the leaf in boiling water, then add iodine into it. Next page number 71. If the leaf contains any starch, it turns blue to black when iodine is added to it. Starch reacts with iodine. Iodine turns from brown to blue-black when added to starch. If a leaf contains starch, it will turn blue or black when iodine is dropped on it. This shows that the leaf has been photosynthesizing. If the leaf has not been photosynthesizing, it will not turn blue or black. So here they are explaining you the starch test. Starch is basically the sugar that plants made and it is known as glucose. So what you have to do is that you need to take one leaf that has green color and boil the water. After boiling the water, you need to put that leaf into that water for about 2 minutes, and after 2 minutes what will happen is that leaf will become soft. Then you need that leaf in alcohol, and alcohol can be nail polish removers. As you know that alcohol is used to remove the nail polish, so it will remove the green color from the leaf. But before adding the alcohol, you need to put out your flame because it burns vigorously when it is heated. Third point is you need to take out the leaf which has no color on it, and then you need to add iodine onto that leaf. Iodine is basically brown in color, so if the plant is photosynthesizing, you will see a color change, that is blue or black. If the plant is not photosynthesizing, then there will be no change in color.
Do you know that leaves of plants produce glucose through photosynthesis? The excess glucose is then converted into starch, which is the reserved food material in plants. Let us do a small experiment to test the presence of starch in leaves. We need a leaf, boiling water, alcohol, iodine solution, test tube, spirit lamp. Take a leaf from a plant that has been exposed to sunlight for several hours. Place the leaf in boiling water. This will kill the leaf, disrupt the cell membrane and soften the cell walls, allowing the iodine to enter the cells and react. Now put the leaf in a test tube and cover it with alcohol. Keep the test tube in the boiling water. The chlorophyll slowly dissolves into the alcohol. Note that alcohol turns green. Removal of chlorophyll will help us observe the color change of iodine clearly. Place the bleached leaf in a petri dish. Pour iodine solution over it. Note the color of the leaf. It changes to dark green. This is because iodine combines with starch present in the leaf and forms a violet-colored complex. This shows that starch is present in leaves. Now cover a leaf with a black paper for a couple of days and do the same test. See if it still has starch.
Next topic is testing the equation. So what the equation they are talking about is the equation of photosynthesis, which involves the carbon dioxide and water in incorporating sunlight, and it will give you glucose and oxygen. We are going to test each and every material that is needed for this experiment, whether the products are produced or not, or whether the raw materials are needed or not. Starting the equation, Using the starch test, it is possible to show that carbon dioxide, light and chlorophyll are needed for photosynthesis and that oxygen is produced. First is oxygen is produced. Leave the apparatus shown alongside in a well-lit place until the test tube is full of gas. Test the gas using a glowing splint to see if it is oxygen. What you need to do is you need to set your apparatus exactly like shown in the picture drawn on the page 71. Then what will happen is that after some time, if the plant is producing the oxygen, the small bubbles will be produced on the test tube and the gas will be collected on top. Second is carbon dioxide is needed. Soda lime absorbs carbon dioxide. If soda lime is put into the plastic bag and tied around a leaf, the leaf will get no carbon dioxide. After a day, the starch test will show no starch is present in the leaf. How you are going to see if the carbon dioxide is required for the plants or not? You need to add the soda lime. What you have to do is that you need to put some soda lime into the plastic bag and tie that plastic bag around a leaf. What will happen is that after some time the leaf will get no carbon dioxide, and then you have to conduct the starch test. That is, you need to put iodine onto that leaf, and what will happen is the plant is devoid of carbon dioxide, so there will be no change in color, which will prove that carbon dioxide is required for the plants.
Starch is eventually formed as a reserve food by plants at the end of a photosynthetic process. In this experiment, we'll demonstrate through a simple experimental setup that gaseous oxygen is evolved as a byproduct during the process of photosynthesis in green plants. To perform this experiment, you'll need the following: a large beaker filled with water, a short-stemmed transparent funnel, a medium-sized test tube of about 20 ml capacity, a few twigs of an aquatic plant such as hydrilla, chara, elodea, etc. To begin the experiment, take a few twigs of the aquatic plant and insert the cut ends of their stems into the tube of the funnel, like so. Quickly but carefully immerse the funnel into the bottom of the beaker containing water, as seen here. Now fill the test tube to the brim with water. Hold the mouth of the tube with your thumb and invert the tube into the beaker and let it sit over the tube of the funnel as shown in the video. The setup is now complete. You may also add a pinch of sodium or potassium bicarbonate into the beaker to kickstart the photosynthesis process. Place the setup in sunlight. A few minutes after exposure to sunlight, you'll notice tiny air bubbles evolving from the cut ends of the stem of the aquatic plant. These air bubbles travel up through the tube of the funnel and eventually collect in the upper end of the test tube. As time passes, more air bubbles collect in the test tube. Leave the setup in sunlight until the test tube is completely filled with the gas. Now, carefully remove the test tube from the beaker while covering the mouth of the tube with your thumb. Remove the thumb and immediately introduce a glowing splinter into the test tube. You'll notice that the glowing splinter reignites with a pop sound. This test confirms that the gas produced by the aquatic plant and collected in the test tube is indeed oxygen. This simple experiment thus demonstrates that oxygen is evolved during photosynthesis in green plants.
During this practical, we are going to see whether carbon dioxide is necessary for photosynthesis. Now what we need is we need 2 destarched pot plants, and to do that we need to place these pot plants into a dark cupboard for 48 hours. Then test one of the leaves out of each of these pot plants to make sure that all the starch has indeed been used up and that there is no starch present in the pot plants at that stage. Then you place these pot plants into a bell jar. Use pot plants that are more or less the same size. In the control, we have soda lime in the funnel, and in the bell jar, sodium hydroxide in the beaker, and both these absorb all the carbon dioxide and make sure that no carbon dioxide actually enters the bell jar. On the other side, we have the experiment. In the funnel, we have normal chalk, and then inside the bell jar, it has water the little beaker has water, but it could also contain calcium carbonate, so that we do have carbon dioxide available so that this plant can indeed photosynthesize. Now we place these 2 bell jars into the sunlight for about 48 hours, and then you are ready to test for the presence of starch in both of these pot plants. The result of one of the leaves taken from the control, we will see that the leaf stays a yellow-brown color, which indicates no starch is present in the control. But in the experiment, we see that the leaf has turned to a blue-black color, indicating the presence of starch. Our conclusion will thus be that carbon dioxide is indeed necessary for photosynthesis, because we saw that starch is present when carbon dioxide.
Moving on to page number 72, light is needed. A piece of foil wrapped around part of a leaf will deprive it of light. After a day, the starch test will show that no starch is present in the parts of the leaf covered with aluminium foil. What you need to do is that you need to take a piece of leaf, and you need to cover a small space with the help of aluminium foil. Aluminium foil will make the plant devoid of light, and after a day, what you have to do is you need to conduct a starch test. How? By putting some iodine drops onto the part of leaf that was covered with aluminium foil. There will be no change in color, and you will see that light is essential for the plants to carry out photosynthesis. Next is chlorophyll is needed. A variegated leaf has no chlorophyll in the white parts. Our starch test will show that starch is only present in the green part. What you have to do is that you need to take the leaf that are yellowish or even whitish in the color, and then you need to conduct a starch test on that white part. There will be no change in color, and you will notice that chlorophyll is required. Did you know the rate of photosynthesis in a variegated leaf is not as fast as in all green leaves of the same size? It is because the variegated leaf have less amount of chlorophyll, and chlorophyll is the substance which gives the plant and the leaf the green color, and which are responsible for absorbing the sunlight. So that's why the rate of photosynthesis will be very low. Next is a variegated leaf can be other color as green and.
Begin the experiment by placing the potted plant in the dark for about 2 to 3 days. This step is essential in order to rid the plant of any starch that was formed in the leaves prior to the start of the experiment. At the end of this time period, remove the potted plant from the dark. Cut a piece of black paper like so, and secure the cut end of the paper using a sticky tape. To make this experiment a bit more interesting and fun, I have carved out my initial, which is the letter T, as well on the black paper covering the leaf. Cover both surfaces of one of the leaves using this piece of black paper like so, and secure the paper on the leaf using some paper clips. Now what we can see here is that these parts are exposed to light, while the parts of the leaf covered by the black paper will continue to remain in the dark. Leave the potted plant in sunlight for several hours. Then, detach the leaf from the plant and remove the black paper from the leaf. Now, introduce the leaf into the test tube containing the alcohol solution. Place the tube in the boiling water bath. If you're using an open flame such as spirit lamp or Bunsen burner for your water bath, then make sure to turn off the burner flame once the tube has been placed in the bath. This is to make sure that the alcohol fumes do not catch fire during the boiling process, which is a huge possibility considering the highly flammable nature of alcohols. Alternatively, you may use a hot plate for boiling like the one I'm using here instead of an open flame. During this boiling step, chlorophyll pigments in the leaf are decolorized by the hot alcohol solution, resulting in a bleached leaf with pale white color. This step is necessary for a better visualization of the iodine starch reaction on the leaf surface in the later part of this experiment. Once bleaching of the leaf has been accomplished, remove the leaf from the alcohol using a forceps. Briefly place the leaf for a few seconds in the hot water bath in order to soften the leaf. Transfer the leaf in a petri dish containing iodine solution. After a couple of minutes, you will see that the exposed parts of the leaf not covered by the black paper turn blue-black in color in the iodine solution. The part which was covered by the black paper, however, do not show any significant change in color except for a faint brown iodine color. This is a negative test for starch and thereby photosynthesis. This simple experiment clearly demonstrates that light is an essential external component for photosynthesis to take place in a plant.
In this practical, we will determine whether chlorophyll is indeed necessary for photosynthesis. For the practical, you would need a de-starched leaf which is variegated, which means that there are parts that are green, which contain chlorophyll, and other parts, if you look on the margin here, you will see that no green pigment occurs in that leaf, and that's why it is colorless. Now, what do we need to do to de-starch this pot plant? We would have to place it into a dark cupboard for about 48 hours to ensure that all the starch that has been produced previously will have been used up, even the parts that would maybe be in the non-colored parts of the leaf. Once we have left this pot plant outside for a few hours, I will then pick one of the leaves and I will test for the presence of starch as shown previously in the first practical demonstration. Now, once we have done that and we have tested for the presence of starch and we have added iodine solution to the leaf after the test for starch, we will see that the parts that did contain chlorophyll has now tested positive for starch. But on the margin, where we see that no chlorophyll was present, we will see that the iodine solution has stayed its brownish color. The middle of the leaf, if we compare it to this leaf, shows that the parts that contain chlorophyll have turned to a darker color. Now, I have placed the iodine solution onto this leaf, and the longer I leave it, it will actually turn even darker. So we can see a definite difference. The longer the iodine solution stays on that leaf, it will turn even darker to a blue-black color.
White. Moving on to page number 73. What happens to the glucose? As you know that plant make its food which is known as glucose, and it uses that glucose to conduct like respiration and photosynthesis. What happens to the rest of it? The glucose that is left off after being used. So reading the topic, glucose is a type of sugar. It is the food made by the plants during photosynthesis. Our plants can use the glucose it makes in many ways. First way is, some of it will be used immediately to produce energy. Respiration takes place inside every living plant cell and all the time, even at night. What they are explaining you here is that some of the left off glucose is used to carry out the process of respiration. Respiration takes place at the night when the plants give out carbon dioxide and they take in oxygen. Next point is, some glucose will be changed into the starch or oil and stored in roots, stems, seeds and fruit. When it is needed, it is changed back into sugar. So what they do is that they store some of the glucose in the parts that does not require that much glucose, that is roots, stems, seeds and fruits. And when the months of autumn arrive, plants uses that stored glucose. Third point is, some glucose is changed into cellulose to make new cell walls. Cellulose gives a strong outer coat to the plant cells. It helps the plant support itself. So what happens is that some of the glucose is also used to make the cellulose, which is the material to make cell walls of the plant. Cell walls is basically the outer covering of the plant cell. It's a rigid outer covering which gives the plant its proper shape. Fourth point is, some of the stored glucose is joined up with the minerals from the soil, like nitrogen. For example, it is joined up with the glucose to make the proteins. Plant need proteins for the growth. Important did you know block? To make the chlorophyll, magnesium combines with the glucose. Magnesium comes from the soil and is taken up with the water by the roots. Glucose, if it mixes with the nitrogen, it makes the protein, and if it mixes with the magnesium, it makes the chlorophyll.
So there's a lot of information on that page in your textbook. Okay, but what happens to glucose that is formed in photosynthesis? What happens? I've summarized it into 3 possible fates. Alright, just like choosing your fate. Alright, so the glucose can be either immediately used by the plant, it can be stored for a later time, or it can be transported to other parts of the plant and then used there or stored there. Alright, so there's three possible fates. First fate, it is used. Now the primary function of glucose, if you remember, glucose can be used for producing energy through cellular respiration, which takes place inside your mitochondria. Okay, hope you're making those links. Now glucose can also be used to make other food substances. Firstly, glucose can be used to make proteins. If you combine glucose with something called nitrate, it will make proteins. We can also use glucose to make fats. And that is why glucose can be used to actually make new cells, new protoplasm for the cells. Right, so the plant makes glucose and uses the glucose to produce energy and to make new substances and new cells. Now the plant doesn't need to use up the glucose immediately, it can store glucose. And the primary way, or the main way that glucose is stored in the plant is as starch. It can be found in the leaves. Right, that's why when the plant photosynthesizes, we can test the leaves for starch. So glucose can be stored as starch. But glucose can also be stored as protein and fat. So what happens is that the glucose that the plant produces is used to make protein and to make fats and is stored in the plant in that way. Alright, so for example, in your seeds, right, like your nuts and stuff, that's actually a storage of fat, right, because nuts contain fats and proteins. So the plant makes the glucose, uses it to make fats and stores it inside seeds, for example. Now finally, glucose can be transported from the leaf to other parts of the plant, and it is usually transported as sucrose. So what happens? Glucose is made, glucose is used to make sucrose. Alright, if you remember correctly, sucrose is made of glucose and fructose. Alright, so what happens is the plant makes glucose, the plant uses glucose to make sucrose, and then sucrose is transported from the leaf to other parts of the plant through the phloem. Alright, so that's a summary. Glucose can be used, stored, or transported. Alright, we are done for today. Now, it shouldn't have taken you too long. If you have some extra time, first start on the worksheet and finish it up. If you have some extra time, you can use it to also work on limiting factors, to revise what that means, because I think last week's content was a lot heavier. So, use the extra time that you have to revise some of that as well. Alright, see you on Friday.